Welcome to QuickSci. Today, we're going to learn about the steps of the scientific method in detail. Remember the steps of the scientific method. They are observation, ask a question, research, form a hypothesis, experiment, analyze data, conclusion, and share your results. Let's begin. The first step is observation. This is the step of the scientific method in which you use your senses to collect information from your environment. Your senses are hearing, seeing, tasting, smelling, feeling. We use our senses to make observations every day. Even right now, you're making an observation. You are using the sense of hearing to listen to my voice as I speak, and you are using the sense of sight or seeing to watch this video. Step number 2, ask a question. After you have made an observation, usually a question comes after. You need to ask a testable question, a question that enables you to carry out an experiment to find the answer. The question must be about changing one thing in the experiment to see how it affects something else. In other words, it must be measurable. Example, does the amount of sunlight affect plant growth? That is an example of a testable question. Step number 3 was research. This simply means that you are using resources to gather information about topics or specific questions that we are trying to answer or that you are trying to answer. When conducting research, it's always a good practice to use credible resources. Step 4, form a hypothesis. A hypothesis is a testable prediction that you will use in your experiment to prove or disprove it. You will use your experiment to determine if your hypothesis is valid or invalid. A hypothesis is an educated guess. Educated because you did your research by gathering information to make a prediction, and you are guessing what is going to happen in the experiment. Your hypothesis also contains two variables, an independent variable and a dependent variable. Step number 5 is experiment. This is when you conduct tests to determine if your hypothesis is valid or invalid. The experiment contains two groups, an experimental group and a control group. In the experimental group, you have your variables, independent and dependent variables. In the control group, you have your control variable. The experimental group is the group in which the scientists carry out the tests, and the control group is the group that is not being tested. This group is actually separated from the test or any activity taking place in the experiment, meaning the control group, nothing is done to it. That is where you have your control variable. Step number 6, analyze data. After you have conducted your experiment, you should have observed or recorded some results in the form of data. You are going to analyze your data to notice any patterns or use it to gather knowledge to determine if it agrees or disagrees with your hypothesis. This data will be used to determine if your hypothesis is valid or invalid. Step number 7, conclusion. At this stage of the scientific method, you will be reporting whether or not your hypothesis is valid or invalid based on the data that you collected. So if your data matches your hypothesis, then your hypothesis is valid. If your data does not match your hypothesis, then your hypothesis is invalid. Step number 8, share your results. At this final step, you are going to share your results with the scientific community, and everybody else will probably carry out that same experiment to see if they get the same results and to see if they agree with you. That's it for today. Stay awesome and have a great day. Goodbye.